focus on the development of, as Peter said, a whole series of new types of nanotechnologies that span molecular diagnostics, new types of materials for in vivo imaging, tools for genetically manipulating cells and making intracellular measurements, and a variety of new types of approaches to doing drug delivery in an area that we're really excited about is gene regulation therapy. And the long haul of what I'd call the modern age of nanotechnology started about 15 years ago or so with the announcement of the National Nanotechnology Initiative. And to me, it's really remarkable how fast things have been developed. I think a lot of people get very frustrated with timelines and start asking, when are we going to see the benefits of this? But you have to remember that in terms of commercial development, in terms of technological development, this timeline has been extraordinarily rapid. I mean, it's amazing that we actually have a large number of things that have entered the commercial market and are making a difference, certainly on the diagnostic front, and I think very soon on the therapeutic front as well. And so at Northwestern, we have a variety of examples of that, ranging from the Verigene system, which is a molecular diagnostic tool that was invented by our group and then commercialized by a company called Nanosphere over the course of the last 12 years. It's a tool that's used for point-of-care medical diagnostics, primarily genomic detection, but also can be used for high-sensitivity protein-based detection as well. And then we have a whole series of research tools that have been developed for studying many aspects of biology, cellular biology in particular, including some of the high-resolution lithographic tools that have come out of this particular effort. Within the current CCNE, there are a wide variety of materials that people are trying to study and take advantage of in the context of both diagnostics and therapeutics. And today I'm going to tell you about one that we are particularly close to, and that's what we like to refer to as spherical nucleic acids. And these are structures that actually have been around for about 15 years. We coined the term about 2 years ago, though, because it was really kind of a paradigm shift in terms of the way we thought about these structures and why they're so interesting and why they're so potentially important in the area of cancer research and the development of diagnostics and therapeutics for treating and studying a variety of cancers. These are structures that are typically made by taking a nanoparticle and developing chemistry to interface oligonucleotides with that particle, effectively using the particle as a template to arrange the nucleic acids in a spherical form. And when you do this in cartoon fashion, you can see the way we envision these, a high-density array of oligonucleotides coming off the surface of the particle, densely packed and highly oriented. That turns out to be really quite critical in terms of properties and how we use those. Not so far from reality, if you look at the EM images of these types of structures, the uranyl acetate stained images, you can clearly see the gold nanoparticle core and the oligonucleotide corona surrounding it. And the thickness of that shell depends very much upon the length of the sequence that we use. And so we can adjust that, and what you end up getting is a structure that's highly synthetically programmable, a structure that can be used for a variety of different types of purposes, but a structure that has properties that are actually quite a bit different from both the oligonucleotide and nanoparticle from which they derive. And that's one of the things that we're taking advantage of here, the fact that you can create a new form of matter, in this case, a new type of structure that has, first of all, very interesting properties from a scientific standpoint, but properties that enable a lot of important technologies. So in the case of a gold particle, the way these are made is we typically take an oligonucleotide with an alkyl-thiol N group. We slowly introduce the two, and then we add salt to the solution. And that's important because the oligonucleotide's a polyanion, and the salt screens the repulsive interactions, allowing you to pack the oligonucleotides on the surface of the particle, having them stand upright to create this very dense shell that gives us then the tailorability that I'll be alluding to. And so we now, actually in very large quantity, at a company we started called Therapeutics, they're using these at the 600-liter scale, preparing them for clinical trials. So this is something, a nanotechnology that is not only interesting, but one that is highly scalable. Now, why are we scaling it? Well, as I said, these structures have properties that are very different from the nucleic acids and nanoparticles from which they derive. And to coin the term, we're looking back at this, asking what's special about these nanoparticle conjugates for quite some time. And it told me after when we started that really what's special about them is not so much the nanoparticle core in terms of the properties I'll be alluding to, but the arrangement of nucleic acids on the surface of the particle. What that leads to are particles, if we compare these on a sequence-for-sequence basis, linear nucleic acids and spherical nucleic acids, structures that bind to complementary nucleic acids 100 times sequence basis. Melting properties, these exhibit these first-order like transitions, almost very cooperative transitions. You can get them down to a single degree, and that has to do with, again, the high density, the high local concentration of the DNA, and also the high local salt concentration. I'll come back to it. A very broad transition. Who cares? It turns out the breadth of the if you use these types of structures, talking about linear or spherical labels in biodetection, is really important. It dictates your selectivity, your ability to pick conditions where the perfectly complementary is bound and non-complementary. When you get into the fact that you can put in lots of different materials, and we'll talk about what they are in a little bit, a lot of progress in the last 6 months in this area, plasmonic, catalytic, luminescent properties. You can even have hollow architectures if you'd like. The physical type of structure. And that's the basis for a wide variety of probes used for medical diagnostics systems that I'll introduce you to. But here's something that really, in this into this phase of the CCNE, we did a Friday afternoon. You're always taught that linear nucleic acid enters repulsive interaction between cells. That's right. Use transfection to effectively clean up the. This is better than any, and there's a reason for the three-dimensional structure of the nucleic acid is not a receptor, which facilitate the uptake of these particles via catalytic endocytosis. And we've worked out the pathways for those of you that are interested in this. There's a paper in PNAS that goes into this in great. But they exhibit almost no immune response. I'll show you some data to that, whereas these, of course, exhibit a very large immune response. The cell does not want nucleic acids in. And they have a resistance to nuclease degradation, which means they last a lot longer inside cells. And the reason for that is the density of oligonucleotides, which sterically inhibits nuclease activation in certain cases, but also the high local salt concentration is incompatible. Think of this as a dense network of nucleic acid on the surface of the particle. It's incompatible with a lot of enzymes, not all, but a lot of enzymes that cannot function in that environment. So we have now gone through and developed a wide class of spherical nucleic acids, taking this really to as far as we can go with it. We use lots of different types of materials, but really important because if you're thinking about therapeutics, for example, it'd be nice if we could go with that have been used, for example, with FDA-cleared drugs. We have cross-linked versions of these structures that are hollow. So hollow SNAs exhibit many of the properties that I've alluded to. Because it's the dense shell of oligonucleotide that gives them those properties. Micelle-type structures, polymer micelle architectures, silica can be used as a network to hold the oligonucleotides together. Metal coordination polymers, gobstopper particle, where you've got nucleotides cross-linking with metal nodes, so that if this, for example, it's constantly releasing acid or acid that can be there, and even liposome-type. It turns out the oligonucleotides, because of their negative charge, will stabilize structures that ordinarily are not stable. Very small diameter liposomes, so that you can make structures in the right size regime here, 25 to 50 nanometers in size, which turns out to be optimum in terms of many of the properties that I've been talking about. So the gold cells are a lot of commercial systems, over 1,200. There are which are very similar described with short oligonucleotide high nucleic acid. It's an interesting structure. It's designed to enter cells like a normal spherical nucleic acid, and once in cells, it can recognize a target that we've predetermined ahead of time, an A sequence, for example, and if it's present, and because it has a greater overlap than what we call the flare sequence, that's peeled off, moves away from the gold nanoparticle, and fluorescence is turned on. Now, if you think about that, that's pretty powerful, because now what you can do is you can based upon differences in content demonstration in Temecula where they've got 70 people that do nothing but crank out nanoflares and a sales force that sells them all. And they started out 18 months ago with this. Have over 1,200. Literally every week, if you go check the Millipore website, there are really 50 new errors added to catalog. This is going to open up a whole new front. Also, the ability to begin to create diagnostic tools, sort and pick out cells, differences in genetic content. We're going to talk tomorrow, for example, on picking out breast cancer in a population of healthy cells. And not only can you pick them out and detect them, but you can isolate them and study them. So if you want to know, for example, how do those cells respond to therapeutics on a personal type of basis, that becomes possible here because you're not killing those cells. And then finally, use to take cells, feed them, focusing on different types of drug. It's asking which one causes an up or down. So, based on the research, powerful, we hope, on the medical diagnostic front. That story has diagnostic. As I said, a high sensitivity sold by nano assays and today, and that is how you can move from diagnostics to therapeutics and be drugs. And this large amounts of genetic material into cells. To create new types of therapeutic capabilities. When we discovered that we get these types of particles in cells, immediately began to think about the fact that it might be important in therapy. Since we're kind of at the different pathways that one might engage with, different types of techniques one might take advantage of. Antisense, where we soak up RNA, that's strategy here. MicroRNA, where we either deliver or soak up microRNAs. siRNA, where we can deliver. We can do that with systems because regardless of your single-stranded or duplex form, they go. A recent one, which is really exciting, is going to become, I think, the subsequent phases of all this, is immunoregulatory. It turns out these structures are taken endocytosis, located in the endosome predominantly. The endosome breaks and a small fraction of 17% or so end up in the cytoplasm. Those are the structures that are actually active for these types of pathways. But for the immunoregulatory, where you're trying to engage toll-like receptors in the endosome, it's phenomenal. The vast majority of your drug and the is extremely effective at engaging these types of receptors. We're finding between 1,000 times more effective than commercially available linear nucleic acid constructs. So it's kind of exciting. This is a that actually took us quite some time to do. It's extremely important, and it really to the power in nano, I think, in a very convincing way. If you look at this linear nucleic acid with a fluorophore, you treat C166 cells with these. You can see with the naked eye there's no appreciable uptake. That's basically teaching us what we already know. Linear nucleic acids don't go into cells. You take that, arrange it on the surface. So it's actually 100 up here. Look at the cells, the constructs. So not only they're taken up in very large quantities, are still greater. The plasma is with these types. You can get not going to be the basis. You have to deal with the toxicity of those types of materials. So very for therapeutics. This is also kind of striking. If you look at the up and down regulation of different types of genes within that cell line, and we've looked at a variety of cell lines in this regard, and all exhibit qualitatively the same type of behavior. If I look at a Dharma effect delivered construct, how many genes are up and down regulated? About 427 in this case. This is using Illumina heat maps. I take the spherical nucleic acid, identical sequence, again, 7. And they almost all have to do with endocytosis. So not only do these go in large quantities, they go in a very stealth-like way. And we think that's going to be very good in terms of creating ultimately therapeutics that don't have a lot of collateral damage and off-target types of effects. And that I'll come back to that theme a little bit later on as well. They go into lots of different cell lines. That's good and bad depending on what you want to do with these. But we have yet to find cell lines other than mature red blood cells that don't pick them up to a significant extent. They're picked up at different levels of extent depending upon the different types of cell lines. For example, keratinocytes pick them up extremely well. For that reason, you can actually create topical types of therapies based upon these. They will also do all sorts of interesting things like penetrate tissues and tumor masses and go right to the core in certain cases. And that made us think that we might be able to create both locally delivered and also systemically delivered therapeutics based upon these structures. How do they go in? I'm going to make, again, long story short, scavenger receptors, in fact, a specific class A scavenger receptors, recognize spherical nucleic acids, whereas they don't recognize linear nucleic acids very well. They localize them on the cell membrane and then dump them into caveolae via caveolin-mediated endocytosis. How do we know that? Well, if we independently knock down scavenger receptor class A type structures or inhibit, we see a significant reduction, if not a prohibition of uptake. And if we knock down caveolin, we see a reduction in uptake. And that's been worked out, but you can then track the movement of these types of constructs because you can follow the gold core. And we use that gold for a variety of purposes, both for imaging, but also for doing mass balance as determined by things like ICP mass spec. So what's the prospect here? So if you think about this, we began to think, look, we have a different mantra than the rest of the world and the rest of the field. Almost everybody in the whole gene regulation field goes down this left-hand path. And I'm not saying it's not a good path. There are going to be lots of good things that come out of that. But you're almost taught to don't even consider going down the other path. That is, if you have a linear nucleic acid or a nucleic acid therapeutic, you need to complex it in some sort of polymeric transfection vehicle. And lots of good things have been done going down that particular path. But we're coming along saying, look, when you have this type of structure arranged in this type of format, you get a single entity agent that's naturally recognized by cells in certain tissues and taken up very readily without the need for a transfection material. And that points to a lot of opportunities. First of all, I've shown you that we don't see significant immune response or significant toxicity. I'll talk about that in greater detail in vivo models. But also much longer construct lifetimes because of this ability to resist degradation. So that means we get much longer therapeutic effects, both in the context of in vitro and in vivo delivered systems. And so we began to work with Alex Steg through the CCNE to begin to look at the development of new types of therapeutics for a wide variety of diseases, in this case, glioblastoma multiforme. I think this audience knows this is an absolutely terrible disease, one with an awful prognosis. If you look at the median survival, 15 months, the 5-year survival is less than 5 months. Unfortunately, if you get this disease, it's almost a death sentence. And not a whole lot has been done in terms of giving folks that have this disease much hope. Well, Alex has been working in this area for quite some time, and trying to get over these types of problems. The standard of care is you always have to take out the tumor. And then there's a combination of radiation and chemotherapy. And this is what ultimately kills the patient, high propensity for tumor recurrence. And the therapeutic strategy failure is due to three things. One is the inability to get drugs across the blood-brain barrier. That presents a challenge. The genetic heterogeneity of the disease. So it moves oftentimes with the therapy. And then a severe resistance to apoptosis. So Alex has been studying a pathway involving BCL2L12 and believes that it is a very good target for potentially treating GBM. And the basic idea behind this is that this is a gene that's overexpressed in GBM tumors. And that if knocked down, you can do two things. One is you can facilitate apoptosis, but also you can make the cells susceptible to treatment with a variety of chemotherapeutics, including TMZ. And so this became a reasonable target to think about using the spherical nucleic acid platform, and first in a cellular model, an in vitro model, and then potentially in a in vivo model. And so we began to look at this, and he looked at tumor neurospheres. These are clusters of cells that mimic in vitro how these types of tumors behave. They're notoriously difficult to transfect, but what we found with these CY5 labeled spherical nucleic acids, they go not only on the periphery of the tumor noosphere, but they go right to the core. So they're able to move through these types of materials and cell clusters and move from cell to cell to cell and eventually find the interior structures as well. Which, again, if you're thinking about developing a therapeutic, a real therapeutic for the disease, that was a very promising observation. If we look at, again, the experiment that I kind of showed you with the epithelial cells now in the context of RNA, but also these types of cells, if you look at glioma cell lines, in this case treated in vitro, this is a problem if we do CY5 labeled siRNA, no significant penetration. That's what you'd expect. You don't see any red dye. You see the CY5 labeled siRNA SNAs, and they go in quite nicely. So at the cellular level, things look extremely good. We know that even with these types of cells, they're taken up by scavenger receptors. If we use inhibitors like poly-I or fucotin of these class A scavenger receptors, you see significant decreased uptake of these types of particles. So we're pretty sure that just like with the epithelial cell studies, these types of structures also rely on this class A scavenger receptor mediated endocytosis. We've begun to build in vitro models of the blood-brain barrier, asking, would it be possible for these types of constructs to move from brain microvascular endothelial cells across a semi-permeable membrane, PET, that's got collagen-filled pores, to the astrocytes below? And the answer is, at least, and this is one of the ones that's typically, none of these are really that good in terms of perfectly modeling the real deal system, the in vivo system, but this is the best that we can do. And this made it look like, or at least suggested that it might be possible if we began to use these types of structures in vivo for them to cross the blood-brain barrier. We down proteins and where we're looking at lines, human tumor neuro to glioma cells. We target L12 with the SNA, and we measure by real-time the MR. And what you find that you can get very good knockdown with the properly. And the persistence is pretty remarkable. If we did this with agent, it would be about half that. We think that, again, has to do with the lifetime of the nucleic acid in the context of the spherical nucleic acid. Linear form. I think important is that the L2L12 knockdown. So if we look at this, the BCL2 targeting SNAs increase effector caspase activation in response to TMZ treatment. So that's the sensitization to the chemotherapeutic. And they also increase transactivation of P53, phosphorylated P53, and downstream P53 target P21 in response to doxorubicin. So this is telling us that the cells, once regulated with respect to BCL2L12, begin to respond the way they should to the chemotherapeutics of choice in this case. Next question became, as we move to the in vivo system, would this be able to move through tissue, brain tissue, for example? And there we work with Tom Mead at Northwestern, who's an imaging expert, and we created a new type of SNA construct, and it kind of shows you the versatility of the platform, where we can load it up with gadolinium. And we've got a synthetic protocol for doing that. I'm not going to go into that, but we can really load up very large quantities of gadolinium on the SNA. Changes some of the charge and some of the properties, so you always have to go through and do all the controls that we did before. But these also are taken up quite well. What's interesting about these is we could begin to look at them not only in cell culture, but also in vivo. And so here's an example where we're looking at a mouse brain where we've done a point injection. And normally, the construct would just be, if it's a linear nucleic acid, localized in the brain. But here you're going to see the path it takes and how much of the brain it actually accesses after 24 hours, and that's going to be in red. And what was remarkable about this, after 24 hours, a point injection with a microneedle accessed about 22% of the brain. And so that suggested if we could get these constructs to the brain, if we could get them near the tumor, we might have a shot at having a therapeutic that could potentially do some good based upon gene regulation. Okay, next question is what happens when you treat these types of, or treat animals, for example, that have tumor neurospheres implanted in them, so basically artificial brain tumors. What's interesting is you can label with fluorophores, and you can use the IVUS system, and you can track by fluorescence. And you can see, in this case, tumors that have been treated with these agents, not only does the construct get to the brain, but it actually localizes, and preferentially localizes in tumor. We think that's due to a leaky vasculature effect, EPR type effect. We have no idea whether that's right or not. That just seems to be the recurring observation. If you look at, first of all, mice that have these surgically implanted tumors, more get across the blood-brain barrier, but in all cases, for the constructs that do get to the brain, the vast majority of the drug ends up in or near the tumor. And we can tell that from these types of studies, whether we're talking about mouse gliomas here or human tumor neurosphere type structures. We know that this is not just the fluorophore ending up there, right? Because that's an important question, because you could be fooled. The gold goes along with the nucleic acid, and the reason we know that is we can take the tissue out and we can stain it with silver. Gold amplifies the reduction of silver 1 in the presence of hydroquinone, and so we can prove that both the fluorophore, which is on the end of the SNA, and the gold is ending up in the brain as well. And we're also looking at ways of tracking the nucleic acid itself. The SNAs do not induce a significant inflammatory cytokine expression in mice. That's good from a toxicity standpoint. And then the second thing I think that's really important is that if we target BCL2L12, is there a substantive effect? Not only do we get the drug there, but is there a net benefit? And we can look at this a variety of ways. One is to look at tumor burden, and what you find is that there's a significant reduction in terms of tumor size for the mice that have been treated with the active spherical nucleic acids, whether we're looking at 21 days or 28 days post-injection. We're going through a series of injections here every couple of days, 1.4 milligrams per kilogram per dose. That actually turns out to be right about the sweet spot in terms of what you want to deliver with respect to nucleic acid. And we can take cells out, and we can measure then whether or not we're not only getting drug there, but are we getting the therapeutic there? Are we getting the gene knockdown effect? And we see by Western blots that is in fact the case. But then the rubber really hits the road when you ask, is there really a therapeutic effect? And if we look at both increased apoptosis and decreased proliferation by ACASPS3 staining and tunnel staining, we see that in both cases we are getting what we would expect based upon knockdown of this gene. And I think fortunate, and it's unfortunate you'd expect to get this type of effect. What we see is increased animal survival. We didn't expect necessarily to see any of this. We have no idea what the active dose should be. But in now many examples, we've done this over and over and over again, in all cases where the animals are treated, the animals with these surgically implanted tumors are treated with these types of SNA architectures, we are seeing significant knockdown and increased animal survival. We think we can get a much larger effect by doing two things. One is optimizing dose, which we're in the process of doing. These are long experiments. You're looking at really 4 years of cumulative work. This was just published in Science Translational Medicine recently. It's the first in vivo use of spherical nucleic acids, at least systemic use. But also when we couple this to chemotherapeutic types of therapies. I'm getting the high sign here, so what I'm going to do is instead of talking about some of the other things, we can also go through microRNA type deliveries. We've shown that we can, as I said, take these types of constructs and put them in creams and deliver them through the skin. Really good for local type delivery systems. We chose the brain cancer example for a variety of reasons. One is it's a relatively low bar to make a difference, right? So we thought this is a great one to try a new type of drug in terms of systemic delivery. It's a terrible, terrible disease, so if you could make an impact there, it would be fantastic. But in terms of long-term toxicity, that's not an issue here, right? Because these patients have a really short lifespan. And so if we could take and significantly increase the length of their lives through the treatment with these types of constructs, that would be a major contribution. Plus, it begins to test the platform, not just for this type of treatment, but for many different types of gene regulation-based therapies. Let me go to, the last slide here, and just acknowledge the folks that have done the work. We, as I said, have a highly collaborative effort. This is my group at Northwestern, and the people that have done the work, Carolyn Coe and Emily Day. Emily's now a professor at Delaware, along with Tim Merkel. And then Alex Stagg, he really is a superstar. He's a fantastic young researcher at Northwestern, the medical school. He's really driving this on the biology side and also on the medical side in his group at Northwestern as well. And then the support, this whole project has been driven from beginning to end from support through the NCI, through the Cancer Center for Nanotechnology Excellence. But I think it's a nice example of again, how basic science and a fundamental understanding of a nanostructure, a new type of nanostructure, has new properties that can engage and lead to new types of technologies that don't just do it another way, but provide really substantive advantages because of those types of properties. I'll stop there and be happy to answer any questions if there are any.
Thank you, Chad. Let's open the paper for questions. Over there.
So, is there a way to remove those nanoparticles from a live animal? Or have you thought about it or done any experiments?
To remove them from a live animal? They will remove if you just wait and collect their feces. You can measure the gold being excreted, and we do that by ICP mass spec. So they'll eventually be processed in the liver, and the animals excrete them. It takes about 3 weeks to get rid of 98% of the construct. The last couple of percent, we don't know how long it ultimately takes to get rid of that, if ever. Well, they eventually will. So what's interesting about these is they have. I took those slides out. That is for almost any of deliveries out there, and it has to do with the fact that they're recognized in almost every cell line out there. So one thing that's not so hot about them is they have a very short circulating half-life because they're absorbed rapidly by all sorts of tissues. If you look at the early stages of biodistribution, you're going to find them in the brain. Small percentage, about 1% gets to the brain. So everything you're looking at here is 1% of the drug roughly getting to the brain. The heart, lungs, these are actually good things in terms of treating a lot of different types of diseases. Spleen, liver, but if you then take a snapshot later on, you'll find that the shift, the biodistribution changes over time, and eventually you'll find the vast majority, like you'd expect, in the liver and spleen.
Yeah, terrific talk. I had always thought that scavenger receptors were primarily concentrated in the liver and maybe macrophages and so forth. Is the distribution determined by the biodistribution of scavenger receptors or by acid accumulation, EPR effect, and things?
You know, he's asking. Can you look at where let me rephrase it. If you can look at where scavenger receptors are expressed, what sorts of cells, what sorts of tissues have cells with those, do you end up getting a biodistribution profile that reflects that? I don't think we know yet, but I can tell you what we do know is that we can take cell lines and tissues that we know express a lot of scavenger receptors. Keratinocytes, for example, have a very high level. And guess what? They take up SNAs better than almost any other cell line that we've looked at. And that's why one of the reasons that both our group and also the company is spending a lot of time on topical applications. There are no topical gene regulation materials, but there are 274 different targets in the skin that one can go after for lots of different types of diseases, spanning melanoma to psoriasis to wound healing to anti-scarring.
Any other questions? I have one quick one. You were talking about this construct being very good to cross blood-brain barrier. Do you understand the mechanism? Do you think it's...
We actually think it's also scavenger receptor mediated. So if I didn't show you, I took the data out, but it's in the paper. If you knock down scavenger receptors, you'll see the ability to cross the blood-brain barrier in the models, in the in vitro models, go way down. So we think that
so
as
it's
well.
not simply because gold is very small. Usually people
No.
say that it's...
No.
So it's
This is not, that's one thing I took out. This is not a size effect, okay? There is a size effect in play, right? You can't use monstrously large particles. Right. But if I take the same particles coated with something like bovine serum albumin, cells won't pick them up. They're recognizing the shell of nucleic acids on the particle surface.
Okay.
So given the promiscuity that you see across all cell types, and I know you're saying this you're suggesting the EPR effect, but I mean, it seems quite localized.
What do you mean?
I mean, you have all different cells taken up except for red blood cells, and it just seems remarkable that tumor accumulation...
It's not that localized. It's only 1% of the injected drug is getting into the brain. 99% is elsewhere. You're just looking at the image of the 1% that gets there. And the 1% that gets there, 2/3 of it ends up in the tumor. So about 2/3 of 1% of the total injected dose ends up in the tumor. The point is, though, that of the constructs that get across the blood-brain barrier, 2/3 end up in the tumor.
So when you look at other tumor models, what sort of preferential...
Oh, it's really interesting. So we look at breast cancer tumor, for example, lung cancer tumors. The tumors will turn bright red because the gold colloid is a red dye too. You can tell with the naked eye that you're getting an enormous amount in those tissues.
But how do you account for that with the epithelial cells and everything else taken?
It's just a matter of rate. It's rate differences. So these things are circulating. Much of the decision is made in the first 15 minutes, and it's which ones can collect and localize for a long enough period of time for uptake, and the tumors end up in all cases taking up more than regular tissues. But I don't want you to think that the vast majority is ending up in the tumor. It's going everywhere, and we take advantage of the genetic knockdown to get the selectivity.
All right, let's thank Dr. Mirkin again.